and
Mhm.
the same thing happened. It said recording in progress. So I thought, oh, great, like we are recording.
Right.
But then it was
That was
actually,
something you didn't want them to do, was it? Or
yeah, and then someone else was recording our meeting, and then this person left with our recording, and I could never trace it back. And I
oh, I
actually have
see.
no recording of my meeting, even
All right.
though, so yeah, it was interesting.
So
And now,
we can admit people. I'm gonna mute myself. So Kira, you're in charge. Just here as a listener. And
I mean, I'll let people join, and then I will wait a little bit. Yeah.
perfect. So
Oh yeah, because you're the host, right? I don't see that. Okay. Yeah.
you should be host now.
Okay, I can help you admit people then. Okay, I think we can get started. What do you think, Francois? Okay. All right, so welcome everyone to this Track the Knot Club. We are very excited to have Dr. Roger Lemon here with us today. Dr. Lemon is FRS Professor of Neurophysiology at the Queen Square Institute of Neurology, University College London, and a Fellow of the Academy of Medical Sciences. His research has focused on the anatomy and physiology of the corticospinal tract and the motor control of the hand in human and non-human primates. Dr. Lemon has extensive knowledge on motor cortex function, sensorimotor integration, and neuroplasticity. And I'm very much looking forward to his talk today. So please take it away, Roger.
Thank you, Chiara, and welcome everybody. Thank you for getting up early, or maybe not quite so early, depending on where you are, to listen to this. I was just discussing earlier the fact that the importance of tractography in the field of human motor and general neuroanatomy of course, it's transformed the field. And then for no other reason, I've become interested in it because, of course, it interacts all the time with what we know from conventional anatomy and conventional physiology. And the big challenge is and has been for some little while trying to understand the cross-validations between these different techniques. Of course, most of the anatomy that I'm going to be talking about comes from the non-human primate. We're fortunate in the motor system, particularly the motor system of the limbs, the correspondence between human and non-human primate anatomy is very close indeed. But obviously there are areas such as speech, for example, where we are really totally reliant on the human neuroanatomy to understand what's going on. Anyway, today is about is a resume of what I think is an update, if you like, on the structure and function of the corticospinal tract. You might think, well, we've surely learned everything we could possibly know about that in the time since it was first discovered back in the Victorian era. But that's actually not true. And as I shall point out, there are some really fascinating questions that have come around when people have started to look in detail at some of the issues. I'll get to that in due course. Right, so now I need to advance the slide. That would be good, wouldn't it? Now, why doesn't that work? Oh dear. Right. Right. So I'm not getting. I am having problems, Chiara, so I can't advance the slide.
I should have tried that before. I
Yes.
see little, I still, can you just, if,
Oh,
yep.
there you go. Something happened there. I'm not sure why that happened then, but
I think you can just click on the slide, it should
oh yeah.
advance.
Okay, it seems okay now. Yeah, good. Okay, so here's a quick summary. I want to talk about structure. I want to a little bit about function. That isn't going to obviously be the headline of the talk. I'm very much concentrating on structure. And of course, one of the initial ideas was that the corticospinal tract was a motor descending pathway from the motor cortex. That's absolutely not true. We now understand it has a widespread cortical origin. It has an equally remarkable range of fiber sizes, which I suspect is reflected in many other central pathways, but we don't have as much detail on many of those yet. And yet it's also characterized by having some of the largest axons in a central nervous pathway. So these are all different facets of corticospinal anatomy. And they certainly relate, obviously, to the function of the system. I want to stress to you today that it's the corticospinal tract has a multiplicity of functions. It's not just a motor pathway. We have a major question around what are the function of the slow fibers that make up the vast majority of this fiber tract, and indeed, again, many other central pathways. Species differences are not only interesting, but give indications as to what functions might be represented in the human corticospinal tract, and not least talking about dexterity and tool use. But I'll touch on those very briefly because most of it is about structure. And I want to finish up by emphasizing a couple of things that where our knowledge of the anatomy of the corticospinal tract has given important insights into human disease, not least motor neuron disease, ALS, and more recently a series of studies I've done with Robert Moorcraft and his colleagues at the University of South Dakota, which has uncovered an unknown and really unexpected feature of the system, which is that it is completely lacking in anatomical somatopathy in its distal parts, that is, the tract within the spinal cord and in the brainstem. So this is the first few slides are just going to be really very basic neuroanatomy, but it's useful to just recap. The first one here is concentrating on the fact that the corticospinal tract originates only from layer 5. All labeled neurons that we know about are present in that layer. And these, of course, project to many targets within the spinal cord, but on the way down there are extensive collaterals. And actually, if you look at if you start from the other end and say let's ask the question, where do layer 5 pyramids project to? Only about 10% of the pyramidal cells in layer 5 actually reach the spinal cord. So the other 90% are going to project to other important sensorimotor structures further rostrally, and including structures like the red nucleus, the pontine nuclei. They're indicated there in green at the bottom of the slide there. That's an absolutely huge projection, and constitutes the loop of information that goes from motor cortex back to cerebellum. So a huge projection to the pontine nuclei, but also to other brainstem nuclei, including in humans, of course, projections to the cranial nuclei associated with speech. Another big projection to the reticular formation, cortico-reticular. But then there are there's 10% of fibers that pass from the internal capsule through the pyramidal tract and into the spinal cord. That 10% is an important figure to remember, as we see when we have a look at some of the sort of numbers around the tractography issues. Now, corticospinal neurons, although they have their name based upon the fact that they are a neuron that projects from cortex to spinal cord, of course, they have many other interesting targets with the collaterals of the same stem axon. And altogether, we call that the connectome. And in primates, at least, the corticospinal connectome is very much concerned with sensorimotor structures. So you get projections to the ones that are listed there: red nucleus, pontine nuclei, cranial motor nuclei, and so forth. But also to sensory structures like the dorsal column nuclei, the dorsal horn of the spinal cord. But outside of that territory, very little. And interestingly, in non-primates, like in rodents, for example, the connectome is much broader. So corticospinal neurons in rodents project much more widely in terms of projections to basal ganglia and to parts of the system dealing with other sensory inputs. So it's clear that during the changes from rodents through carnivores to primates, this connectome has become more and more concentrated on sensorimotor function. I studied for some years with famous anatomist Hans Kuipers, and the slides I'm putting up now are based on his work. And I'll put this one up because it quickly summarizes some of the most important projections that these layer 5 neurons made. This only relates to the projection from the motor cortex. And within the spinal cord, Kuipers identified at least 3 major areas of termination. One, indicated by this white area at the bottom there, involved a direct projection, which is unique to primates, the corticomotor neuronal connection. And that's a connection that's particularly strong to extremity muscles. So in the upper limb, the muscles of the hand and fingers, and in the lower limb, the toes and the foot. The second major projection is to the so-called intermediate zone, shown in green there. So these are parts of the, shown in sort of pinky brown there. And this is the part of the spinal grey matter that doesn't contain motor neurons, but does contain a wealth of different sorts of interneurons. They receive a very heavy projection, cross projection from the motor cortex. And then finally, there's an uncrossed projection, so that these made up of fibers that don't cross in the pyramidal decussation at the base of the brainstem. And this is a bilateral projection and involves the motor neurons and the interneurons that influence trunk and girdle muscles. So this is probably a very much older, evolutionary older projection. It's bilateral, and it's all to do with keeping the girdles and the trunk under control. That's shown there in green. Of course, there are many other descending pathways which I don't have time to look at today, but it's important to realize that, of course, there's a whole wealth of brainstem pathways that, in many respects, parallel the corticospinal pathways, but they're differently organized and have different targets within the spinal cord. So concentrating on the corticospinal system, I've already talked about the widespread cortical origin, always from layer 5, the descent through the internal capsule, passing between the basal ganglia and the thalamus, a major coming together of all the fibers within the pyramidal tract. This is a rather unique assemblage of fibers because it is a pure descending bundle of fibers on the base of the brainstem that decussates after the olive, and most of the fibers cross over, about 10% don't, but the cross fibers then go on, and they project very widely within the spinal cord, right from cervical down to sacral regions of the cord, and as far as we know, they project pretty much to every laminae within the spinal cord, which tells us they must have many different functions. And they include direct monosynaptic connections indicated in this little diagram here, particularly to motor neurons supplying the most distal muscles. The brainstem innervation is a lot to do with facial musculature, particularly the muscles around the corner of the mouth, and of course the muscles of speech, and then hand, fingers, feet, and toe muscles. So I've already mentioned some of these key features, multiple areas of origin. What you see here is a picture of a monkey brain, macaque brain, where a large amount of retrograde trace has been injected into the spinal cord and has been transferred retrogradely into the cortex. And you can see a very wide regions of distribution of the label cell bodies. Of course, they're concentrated here in the precentral gyrus and the motor cortex, but they spread well into the frontal premotor areas. There are areas on the mesial side of the hemisphere, callosal and limbic areas, which include many parts of the parietal lobe, and that you can see also on the lateral surface of the hemisphere, extending into the parietal sulcus. And then there's a bunch of neurons that are buried in the Sylvian fissure and originate from an area called the second somatosensory area. So a very widespread set of different origins, mostly recognized as clearly differing in function. So that emphasizes the point I want to make about multiple functions for this particular system. The next feature that emphasizes multiplicity of function is the range of axon diameters. So there's a 100-fold range of diameters from a fraction of a micron up to about 10 microns in the monkey. In the human, the axons are even larger. They go up to about 20 microns in diameter. All these fibers are myelinated, but as you can see, there is this huge range, and there's also a very, very clear predominance of fine fibers. Very much the majority are small fibers. And the final feature, the projection within the spinal cord, that doesn't just project to motor areas or sensory areas of the spinal gray. It projects to the areas related, for instance, to autonomic function, to in extremely crossed sections of the spinal cord, and regions that deal bilaterally with the control of girdle and trunk muscles. Now, this 100-fold range of fiber sizes is very well demonstrated in the anatomical literature. It's been known for a long time. So in this series of pictures here in A, you see the whole pyramid identified. Here's the medial fissure. Here's the pyramid lying on the base of the brainstem. Here's the inferior olive. If you go up very considerably, this is a light microscope view, and you see the profiles of the myelinated fibers in the pyramidal tract. it's very rare to see perfectly round profiles. We don't know whether that is partly due to the preservation routine, or whether this is naturally. So here's an EM picture of in C, and finally in D we see an example of an oligo dendrocyte wrapping itself around a number of really tiny fibers, a family of very small fibers, less than 1 micron in diameter, which actually make up a very significant proportion of the numbers present. And here's the picture I showed you earlier. This is from a study we did about over 10 years ago now, using both light microscopy which is in green, and electron microscopy from the tissue from the same animals. And you see a pretty close correspondence, except that the EAM does clearly show up a population of very fine fibers that we don't see and can't see under the light microscope. But it also stresses that there are, although difficult to find because there are not many of them, some rather large axons. So first of all, make the point that most of the fibers are less than 3 microns in diameter, but there are some larger ones, and that's blown up here. You can see that there are very small numbers of these because, of course, this was done by a painful process of counting and measuring individual fibers. It took a long time. But even so, we would calculate that in the macaque corticospinal tract, these large fibers, that is 8 microns or bigger, probably amount to something in the order of between 15 and 20,000 axons altogether. So it's not an insignificant number. Now, interestingly, the picture we see from the anatomy is the complete inverse of what we see from the physiology. So people have been recording from these pyramidal tract neurons for ages. It's possible to identify those physiologically by antigenomically activating them from the pyramidal tract, from this a tract which only has these axons within it. It's not contaminated by any other systems. So if you stimulate that system and so-called backfire the neuron, get an antigenomic response, you can, and here's an example in the heavy black image here. Here's a stimulus artifact, and sometime later, this is an antigenomic response of a pyramidal tract neuron. If you measure the interval between the delivery of the stimulus and the arrival of the antigenomic response at the cortex, and you know the length of the pathway between the two, you can work out the conduction velocity of the axon. But the first thing to say is that if you do this and sample lots of neurons, then the picture, the distribution is very much dominated by these very fast so-called PTNs, pyramidal tract neurons. So you see this clustering down here, particularly in the primary motor cortex, M1. You'll see there are lots of neurons with an antigenomic latency of only 1 millisecond, which gives them a conduction velocity of about 80 meters per second, which means they're coming from quite large fat axons. But you'll also see there are very few neurons with very long latencies that ought to belong to the PTNs. There are many, many PTNs which have much finer axons, and that's been a mystery, but has been solved by the realization that most of these studies were done with single microelectrodes that weren't particularly good at identifying the smaller neurons. So we ended up with a picture saying, well, the pyramidal tract is a system that has axons in it from 12 to 90 meters per second and not much more, because we didn't see any or very few PTNs with conduction velocity of less than 12 meters per second, which would be in this from around about this region to the right. So very little evidence for these slow ones. But in more recent years, people started using different sorts of electrodes, particularly very sophisticated multi-contact electrodes. Let's see what I've done with that slide. And that has answered the question of where are all the slow ones. have disappeared to. But before I get on to that, I just want to make it was a sort of classical point that these fast axons seem to show up in particular species, particularly in large species. So the question was, is it just a question of reducing the time delay between cortex and muscle? That if you're a very large animal with a big body, you need to reduce the conduction delay from brain to muscle. So that's why you need large, fast axons. But actually, if you look at that question, you find that in fact, it's true that most axons over 8 microns, so these are quite fat axons, are only found in large primates. That's certainly true. For instance, gibbons, macaques, spider monkeys, humans, good examples of primates with very really quite chunky, large, fast-conducting fibers. But it turns out if you just look at body size alone, this doesn't really correlate with the size of axons. So, for instance, elephants, obviously very large, largest fibers, 8. The domestic cow, 5 microns. And just for completeness, going to the opposite extreme, very small animals all have very small fibers, about 3 microns in diameter. So I've already mentioned that one reason why you might like to have large fibers, that's to reduce conduction delays, but almost certainly that isn't the whole answer, as I hope I showed you in the last slide. We do now know that large fibers tend to be the ones that contribute direct motor non-synaptic connections, CM connections, corticomotor neuronal connections to motor neurons, and they're only found in some primates and in humans, of course. But interestingly, those projections support an important feature of corticospinal function, which is the ability to fire at very high frequencies. So it may be that the fast conducting axons are able to generate these fast conducting messages to the cord, which the thinner ones can't manage. And the other reason is, of course, is to do with the connectome and the branching, the amount of branching that goes on from the stem axon, and that is an important feature in terms of making sure the same signal arrives at all of the targets included in the connectome. And branch failure at collateral branch points is a problem. If one branch gets a signal and the other one doesn't, that's going to lead to errors within the network. So, and again, it turns out that the transmission at branches is more reliable with large axons than with thin ones. However, I want to emphasize that small fibers could be very important because, firstly, they're numerically much more numerous, but also they are actually much more resistant to trauma than are the larger ones. And this is an old slide from the Miami group, which showed that if you look at this section here, is the spinal cord of someone who's had a serious spinal cord injury. And if you look a few segments away, you'll see that the area of the corticospinal tract here in the dorsal part of the lateral columns is very pale. And this is because it's lost all the large fast axons, because these have died. But the finer axons in the lateral corticospinal tract seem to have survived, probably because they're metabolically more resistant to the loss of proper perfusion than the larger ones. So they are important clinically, at the very least, these fine fibers. So I want to just spend one or two slides on the question of why aren't the slow PTNs well represented in the physiology? Because if we don't have them in the physiology, it's difficult for us to ask questions about function. And it became clear really in the last few years that the reason why they have not appeared in the classical physiological studies is that people were using electrodes which were far too crude and coarse to be able to see the tiny potentials that the small bodies, cell bodies of slow conducting PTNs generate. And Stuart Baker in Newcastle led a piece of research a few years ago, which showed that if you use one of these multi-contact silicon probes, you can actually record these antidromic, very slow antidromic potentials. I wouldn't say easily, but certainly with much greater ease than the old conventional microelectrodes. So, for instance, in this slide here, you'll see these waveforms at the bottom here, labeled with either an open or closed circle. So these are antidromic responses from slow pyramidal tract neurons, occurring many milliseconds after the stimulus, which is over here, into the pyramid. So you can find them if you have the sensitive enough recording equipment to do it. And this slide summarizes the results here, showing the fast PTNs which dominate the picture using conventional electrodes, but these the black columns here are the so-called slow PTNs recorded with these multiple contact probes, and they, as you can see here, they have conduction velocities below 12 meters per second, and that would correspond to axon diameters of 2 microns and finer. So it is possible to see them. Unfortunately, it's still an open question as to what all these very, very tiny fibers, amounting to more than 90% of all the fibers in the corticospinal tract, what their function or functions are. More generally, we know that the system does not act simply as a descending motor pathway. It has motor and non-motor functions. The motor ones I won't dwell on too long, because I'm sure they're familiar to most of you, but they include direct, monosynaptic, and indirect excitation of alpha motor neurons, inhibition of motor neurons, control of spinal reflexes, modulation of things called spinal pattern generators, which are important for very basic motor patterns, including walking, swimming, scratching, standing, and so on. And interestingly, they also include the motor aspects of the mirror neuron system. This is a system in which neurons in the brain are active when we observe the actions of others, and we know that this actually activates the same projection, but at a subliminal stage, at a subliminal level, so that the imitated function is not repeated by your own motor system while you're watching it. But let's look at the non-motor ones, because these are actually very interesting. One of the ones that's become more and more evident is the control of afferent input. So think of a descending pathway, particularly from sensory areas, somatosensory areas of the cortex, involved in the control of incoming sensory input, including nociception. A very important function of the system then is the control or gating or filtering, or in some way interacting with the incoming sensory input. Autonomic control, also very important, and long-term trophic effects that these fibers exert that we know very little about, but which are almost certainly important during development and during recovery from injury. A brief section now on species differences. This is a lovely old drawing from Hans Kuipers, where he compared the projections using conventional anterograde labeling methods, which was a neurodegenerative technique, a silver staining technique, which required you making a lesion, then waiting for the fibers to degenerate and then stain the degenerating fibers. And what he saw in these studies was that in non-primates, such as the opossum and the cat, most of the corticospinal fibers terminate dorsally in the spinal gray matter, and there's no projection into the ventral parts of the spinal gray in the ventral horn, the anterior horn, where the motor neurons are located. And the slide also shows that in primates, such as the rhesus monkey and the chimpanzee, there were much heavier projections that spread into this anterior horn, the ventral horn, And these, of course, are the fibers that are going to contribute to the direct motor neuron control from the cortex. So that systematic investigation showed a sort of ventralward shift across this particular set of species, from dorsal to ventral. And later on, as more and more studies were done, it became clear that these direct corticomotor neuronal projections were unique to primates and unique to dexterous primates, so primates that are good at using their hands, making tools, and using tools. But that doesn't undermine the importance of what I was saying about projections of this system to somatosensory systems. In the rodent, for example, shown on the right here, there's a very heavy projection to the dorsal horn. You see, this isn't using now an anterograde tracer injected into the cortex and then revealed to show that most of the terminations from this system in the rodent are in the dorsal horn. Now, interestingly, there's a parallel projection in the primate, and certainly in humans, also to the dorsal horn. Here you see the axons in the lateral corticospinal tract, and here you see the terminations within the dorsal part of the spinal gray. But that projection comes only from S1. So you don't see any projection to the dorsal horn from the primary motor cortex. So you see a very heavy one from the primary sensory cortex. And alongside these anatomical revelations, there are a number of physiological behavioral studies which have shown how important corticospinal projections are for touch and pain function. Most of those studies have been done in rodents, but it very clearly identifies the importance of that projection to the somatosensory system. To concentrate on what happens from the motor cortex, where we can think of the corticospinal tract as its in terms of its descending motor influences. This is work from Robert Morecraft's group, looking at projections from the precentral gyrus coming down, crossing in the pyramidal decussation, traveling in the lateral corticospinal tract and terminating very heavily in two main layers, in two main parts of the spinal gray, lamina 7 and lamina 9. And these different colors indicate the relative number of boutons. They counted the labeled boutons, the relative number of labeled boutons in these two laminae. And I want to draw your attention particularly to these ones here in the C8, T1, C7, C8, and T1 parts, segments of the spinal cord. There's a very heavy direct projection into lamina 9. And this, of course, is where we find the motor neurons belonging to the most distal muscles, the wrist, the hand, the fingers, and particularly the thumb. And this shows a numerical comparison showing that the projection to the intermediate zone, shown in red, lamina 7, is clearly the heaviest, but there's a very significant projection directly to motor neurons. And I won't have time to go into it, but it turns out actually that probably quite a lot of this red stuff is actually terminating on the distal dendrites of motor neurons. So the motor neurons may be located, and they are located in lamina 9, but they have dendrites that spread right throughout the spinal gray matter. And it could be that some of these terminals here are actually on the distal dendrites of those same motor neurons. So they could still be involved in this monosynaptic projection. Just a few words about how this all fits with tractography. And as I said, I don't want to be in any way seen as a critic of tractography, because it has totally revolutionized our visions of pathways in the human brain, and that ultimately is our is what we want to study, not the monkey brain, but the human brain. And it has actually rekindled a huge amount of interest in both the physiology and anatomy of the system. The point I want to make, however, is that the accuracy of the method depends very much on how careful you are with the placement of the seed and target ROIs. And particularly, I come back to this point that you have to recognize that the corticospinal system, if that's what you want to study, does require the seed voxel to be in the pyramidal tract, or even better, in the spinal cord. Because if you go any more rostrally, you are including as corticospinal actually a vast number of fibers that never reach the spinal cord and actually terminate in structures like the pontine nuclei. So remembering that the corticospinal tract is made up of only 10% of all the corticofugal fibers that come down from the cortex, that means that trying to show the representation of the corticospinal tract within the internal capsule, that's a very important thing to try and do. But to do so, you need to get your seed voxel in the right place. If you put the seed voxel any more rostrally, which many studies have done, simply in order to make it easier to get a larger and better representation of where these fibers are running, you are actually including a lot of fibers that are almost certainly not corticospinal. And just to illustrate that in a very crude way, this is a study from Mitch Glickstein, and he did a very simple thing, which was to measure the complete surface cross-sectional area, cross-sectional area of the cerebral peduncle here at the junction between the forebrain and the midbrain. So this dark area here include all of the corticofugal axons that are going from many, many different cortical areas through to the midbrain and brainstem. And he compared that area to the area of the pyramidal tract, which is, of course, at the base of the brain, much more caudally. And basically, this is around about an 8 to 10 times difference in cross-sectional area. And therefore, assuming that the fiber distribution is relatively similar in both, we don't know that, but assuming it is the same, means, underlines this point about only a fraction of the fibers in the peduncle actually reach the spinal cord. Just one slide on, I think it's just one slide on function. Obviously, from a long time back, way back from some absolutely wonderful studies by Sarah Tower, which showed that the one really striking deficit caused by cutting these fibers at the level of the pyramid was the loss of fine finger movements. And that was confirmed by a much later study by Lawrence and Kuipers, did a similar thing, but I would say, that they were able to, with the advent of better neurosurgical techniques, they were able to get nearly all their animals to survive the operation for long periods, and so could study the permanent effects of these lesions, whereas Sarah Tower's animals tended to die very soon after the operation because of all sorts of complications from the making of the lesion. But both sets of studies emphasize the fact that if you cut the corticospinal tract bilaterally, the one thing that you lose completely and permanently is the ability to move your hand in a skillful way and to be able to do things like precision grip. And we now know that the CM cells that travel through the system are functionally organized. They can be shown to be differentially active according to the different sorts of grasp that a monkey or indeed a human is trying to make. We can show that these same cells are active during things like tool use. This is a picture from Atsushi Eriki showing a macaque monkey using a rake as a tool in order to retrieve food that it cannot reach with its arm. It can extend its reach by picking up a rake and pulling the food towards it like a croupier. And so a long time ago, Lawrence and Kuipers attributed the loss of independent finger movements, particularly to the loss of the direct projection, because it was very much more striking in animals that have this CM projection than in those that do not. Now, of course, what we'd like to know is there a CM system in humans? And the evidence comes from two main lines of work. Firstly, of course, using old-fashioned silver staining methods, looking at degenerating fibers after stroke. So there are a number of studies of patients who suffered a severe stroke in the motor area, for example, and then died several weeks later. And then if you looked in the spinal cord, you could see where the fibers from that damaged area of cortex terminated. And here's an example from the 1960s showing in the black stipple the points of which they could see degenerating terminal boutons of the corticospinal system. And you can see that they're located in a number of different areas. This is after a motor stroke in the primary motor cortex. And particularly in this region here, the most lateral part of lamina 9, which is where the intrinsic muscles of the hand have their motor neurons, there's a very strong projection. So this looks very similar to what you can see in the macaque monkey. The other evidence comes from physiology, using non-invasive means of activating these same neurons transcranially, using either a magnet or an electrical device. And if you do that, when you deliver the stimulus, that's in this case using an electrical and transcranial electrical, you see an artifact here, and then a response here around about 20 milliseconds later. And if you look in great detail at this response, I really spread out the time here, you can see that all those responses fit into a very, very tight package. And that is exactly what you'd expect of a monosynaptic connection, because it means that there is no additional time for other oligosynaptic or polysynaptic pathways to be activated. The response is short, but it's also very, very synchronous, because as soon as you introduce extra synapses into the system, it becomes more diffuse and jittered. Just one slide on the larynx, because similar studies have been made, in particular by a group in Spain and Croatia, led by Vedran Delitis, late Vedran Delitis, sadly missed. And what he did was to put very fine wires into a muscle called the cricothyroid muscle, which is an intrinsic muscle of the larynx. And he showed that if you stimulate the cortex directly, this was done during neurosurgical procedures, you get these, I think, see these, this is cricothyroid, CTHY. You see these responses in the recordings, which are incredibly fast and very synchronous, again, suggestive of a very strong direct projection from the motor area, from the laryngeal component of the primary motor cortex to the intrinsic muscles of the larynx. And interestingly, that projection is not found in non-human primates. It's neither anatomically nor physiologically. Okay, rushing on. I'm going to miss most of this, but this is just to emphasize the fact that this direct projection that I've been going on about comes mostly from the bank of the central sulcus. It comes from an area which is called by Peter Strick new M1, because it's evolutionary new. It's only found in non-human primates and humans. There is a projection from the more rostral region, so-called old M1, but this is much less numerous, much less important probably than the projection from the caudal new M1. And to point out that so-called new M1 in humans is almost entirely buried, which isn't absolutely entirely buried in the bank of the central sulcus. So it's not available to a surgeon, for example, who's looking at the exposed surface of the cerebral cortex because it's buried deep down in the bank. So you have to use an invasive probe to actually activate these areas here. Area 4A and 4P, which is the new M1, is a long way down, around about 1.5 millimeters at least from the cortical surface. So I'm going to miss this next slide and finish up by just saying a couple of things about disease and so as to stress the importance of this system, the CM system in particular. In one particular area, motor neuron disease, that's been a great interest of mine. It turns out there's a greater weakness in the muscles that receive the strongest CM projections. And this fitted with the discovery of a thing called the split hand syndrome by Andrew Eisen about 20 odd years ago. He discovered that the thumb and the index finger are always weak, very, very weak in patients suffering from motor neuron disease. And that's probably because they have specifically lost the large, fast projection to these muscles. And interestingly, on the hypothenar side, this is why it's called split hand, on the side of the hand for the hypothenar, the muscles there are much less affected than are the thumb and index finger muscles. So that's been one very important example. So we know that the pattern of muscle weakness in motor neuron disease probably reflects the pattern of corticomotor neuronal innervation in health. And then the very last part of the talk is about, and I hope I've got time to just go over this, Chiara, but one of the things that we got very interested in was trying to understand what is the pattern of fibers within the corticospinal tract. So we know that obviously there are areas of the primary motor cortex. You think of back to the classical work of the homunculus, areas devoted to the face, indeed to the larynx, to the hand, fingers, trunk, leg, toes, and feet. And we were interested to know whether, people have been very interested to know whether this somatotopy, how that related to patterns of motor disability, particularly in patients with spinal cord injury, and in particular, one particular type of injury called central cord syndrome, which is thought to be due to traumatic damage to the very central part of the spinal gray. And the feature of these patients is that they have a thing called man-in-a-barrel syndrome, which means they have a much greater impact on their upper limb function than on their lower limb function. And this was explained classically by a diagram like this. This is from a relatively recent textbook of neurosurgery. The idea was that the leg fibers travel close to the outside of this is the corticospinal tract in orange here. The lower extremity LE fibers are on the outer regions of the corticospinal tract, and the upper extremity are located more medially and around about here. And the idea was that if you had a central damage in the core of the cord, that it would more likely affect the upper extremity fibers than the lower extremity fibers. That was the idea. And what really was interesting was to find out where this idea came from. It came from a neurologist called Otto Förster, who was also a bit of a neurosurgeon. He published this diagram in the 1930s, and it shows a very clear lamination. This cord is upside down, showing that there is arm medially and leg laterally within the corticospinal tract. And that's been reproduced in all sorts of places, including Gray's Anatomy. Here's Gray's Anatomy in 1950 something, I think. Leg lateral, arm medial, trunk in the middle. Now, what is quite remarkable when you talk to experimental anatomists who study the system is they've never seen anything like this in any of the studies that they've done using conventional or even very much up-to-date anatomical tracers. So this is just the same figures again. And I got Robert Moorcraft in the University of South Dakota interested in this question. And what he did was to look at some old papers in the monkey, which showed that if you look in the monkey, if you make one small injection within M1, you see fibers everywhere within the tract, not just in one particular zone, which is what you'd expect if there was a detailed somatotopical arrangement. So, for example, a lesion or an injection in the hand area of motor cortex labels fibers throughout the pyramid. You can see that here and here, not just in one zone. Anyway, so we needed a good test of this, and we wanted to overcome some of these old techniques because not all of them labeled all the fibers in the tract. The fine fibers weren't labeled, and nobody had ever looked at what happened to the somatotopy of the projection from areas other than M1. So here's Robert, and here's the experiment that he did. He basically injected different regions of the primary motor cortex, for instance, leg area, shoulder area, arm area, and asked the question, is there localized or is there widespread distribution of the fibers? The one would reflect clear somatotopy, the other would suggest that that was not the correct picture. That also would expect that if there was no somatotopy, there would be a lot of overlap in the location of fibers labeled from these three different sites. And it turned out that that second conclusion was the correct one. So, for example, here we see what happens after an arm-hand injection in M1, and you see fibers labeled throughout the pyramid up at the top here. And if you go into the cord in the lateral corticospinal tract shown here, you can see that it's not just one region of the tract that's labeled. There are fibers throughout the tract. They're everywhere. And if you compare that with the leg and the foot region, when that was injected, exactly the same regions were labeled after leg and foot. So in other words, it means that the arm fibers and the leg fibers, the hand and the foot fibers are all very heavily intermingled together. I'll miss these next two because that was just a stereological confirmation that this conclusion was safe and secure. We also saw similar overlap between M1 and supplementary motor area. Again, the fibers travel in the same regions, and an overlap between S1, between the arm area of S1 and the leg area of S1. So for every cortical component of the corticospinal projection, there's an extensive overlap and or wide distribution of labeled fibers. And this diagram tries to summarize what we found and shows that in blue and green and magenta, we're showing the fibers that are traveling from arm, hand, shoulder, and leg areas of M1. And you see that because the colors are clearly all intermingled, that reflects the fact that there is no obvious somatotopy within this projection. So how then can we explain the thing that we started studying, which was why do patients with this damage have a bigger problem with their upper limb than the lower limb? And we concluded that it's almost certainly the case that if it's a diffuse injury and goes diffusely through the whole tract, it would produce exactly that pattern if it were the case that the corticospinal projection is much more critical anyway for upper limb rather than lower limb function. And I don't have time, obviously, to go into that evidence, but there are plenty of studies that show just that it's the control of upper limb function that is much better developed, particularly in humans, than lower limb. And so if you're unfortunate enough to have a traumatic injury to the spinal cord, you will get a much bigger effect on the upper limb function than the lower limb function, but that doesn't mean there's somatotopy. It just means that the whole tract is more influential over upper limb than lower limb. Right, concluding slide. So it's a multifunctional pathway. Hope you're convinced of that. The largest fibers make these direct corticomotor neuronal projections, connections, but we don't know very much about the small fibers. While we can show quite clearly that there is a somatotopy within the internal capsule and cerebral peduncle, below there, once you get into the pyramidal tract or lateral corticospinal tract within the spinal cord, that organization disappears. Now, you might ask then, what helps fibers get to where they need to be? How does a leg area neuron know to send its axon to leg motor neurons and not arm motor neurons? And from work in rodents, that is becoming increasingly clear that that is coded by proteins in the growth cone during development that have a conversation with their target motor neurons and presumably are repelled by the wrong type of motor neuron and attracted by the right type of motor neuron. How that applies to primate development is an unknown at the moment. And finally, the absence of this somatotopical arrangement in the human corticospinal tract certainly has important consequences for understanding recovery of function, because we know that, for instance, small lesions don't affect the whole tract. They will leave some fibers intact in what's called incomplete spinal injury, should leave a proportion of fibers representing all types of function, despite the fact that the person has suffered a major lesion. But having this knowledge about the intermingling of fibers should help to understand not only the injury, but also the recovery from the injury. Thank you. I finished.
Thank you so much, Roger, for this wonderful overview. Realizing that we are past 10, I am more than happy to stay a few more minutes if you are available, Roger.
Yeah. I'm sorry. As always, I've overloaded the number of slides. But if there are any questions, I'll be happy to try and give some very quick answers.
Yeah, exactly. So I would encourage anybody who has a question to just unmute themselves and ask. And maybe we can do what we did last time, which is if you stop sharing your screen, we can all show our faces, which helps discussion.
Yeah.
Yeah, and also I said that if people had to leave at 10, they could write their question in the chat. So if we can talk about them, they could watch the recording later.
Just trying to work out where the button is for stop sharing.
There should be a red tab saying stop sharing.
Okay, just a second.
Or in the top of your screen.
Stop
Or
yeah,
the host can.
I got it. We got it. Thank you.
All right. Okay. Does anybody have questions?
I had one. If I can go. I don't see anyone else. So when we work with tractography, we have a few hypotheses about like how the axon are organized. And it seems from previous talk with Kathleen Rockland, for example, we say in tractography, axon should have a straight or smooth path along their length. Like they should not curve 90 degrees for no reason. And then Caitlin showed us sometimes it happens. So like that kind of break one of our hypotheses. And sometimes we say axon diameter should be like a distribution in a small, let's say, voxel. Should be like average plus variance. But then we see, oh no, it's more like multimodal. Like you can have two population of axon with two different axon diameter, like in the same area. And then we say also axons should have a smooth, continuous like axon diameter along their length. Is that one false too? Like can axon like have various diameter along their length? Like I'm just trying to see like if all of our hypotheses get broken one after the other, but what's left at the end?
Yeah. Well, certainly, it's certainly the case that the diameters of wide variation are traveling within the same area of the tract. So when you look at the electron microscope, within one within one field, you'll see many fibers, from the thickest to the thinnest, all traveling along happily together in the same zone. The question of how constant the diameter of any one axon is from cortex to spinal cord is an intriguing one. Obviously, extremely difficult to study. But all I can say is that there are some electrophysiological studies which looked at this question by stimulating the axon of a single cell at many different levels of the cord, and then asking the question, is the conduction velocity the same all the way along? And the answer is it isn't, but it doesn't change that much. It does, of course, tend to slow down the further away from the cell that you go down. But basically, a fast-conducting axon remains a fast-conducting axon throughout its length.
Thank you very much.
And sadly, I don't think there's any information on whether that is the case for the slow ones, but we are beginning to kind of open that particular box and try and find out whether that's also true.
Thank you very much. This answered my question.
I see a question in the chat from Maxime asking, any knowledge on unmyelinated axons and their role function within the CST?
Right. Well, one of the intriguing things about this particular pathway, obviously it doesn't apply to all of them, is that there are no unmyelinated fibers within the corticospinal tract. Even the finest fibers have something of a myelin sheath. May only be one or two lamellae, but they are, as far as we know at the moment, there are no unmyelinated fibers within this system. Obviously, they are going to conduct even more slowly than these very fine fibers that I've been talking to you about. So, that raises interesting questions of function. But I think if we understand that axons may be influencing the spinal cord or their target area more generally, not only through electricity, not only through action potentials, but through trophic influences, involving the release of compounds from synapses and so forth, then one begins to think of these very fine fibers as having very slow but potentially very important functions, but they don't involve rapid transmission of action potentials.
Interesting. Kathleen, yeah, go on.
Yeah, Roger, so listening to you, you're making me think that there must be another difference is when we're in the brain, the oligos, the number of oligos in relation to the axons that they're wrapping will be quite different when you get into the periphery in the spinal cord with Schwann. We have to think of Schwann cells, and there's different ratio. Is that correct, or am I misremembering?
Yeah, I would imagine so, but again, I wouldn't, I don't think there's anything out there. I mean, I think the study that we published with Mitch Dickstein in about 10 years ago was the first EM study of the macaque corticospinal tract, and the human ones that are, of course, much older. So there's a terribly important question that you're asking. And I can't really offer very much in reply. But I say I think it's really intriguing that there are, as far as we can see, there are no unmyelinated fibers, and that a system that has such a huge range of diameters must, by definition, be involved in a huge range of different functions, because the nervous system doesn't work like that. If there's a particular function, then it tends to be constrained within a particular population of fibers of given size and conduction velocity. And so the sort of 100-fold range that has been identified, I think, does tell us that there's a whole lot of things going on that we probably know very little about. And of course, the physiology has tended to distract us to work only on these very big fibers, which are easy to record and exciting to work with, because they clearly represent a means of the cortex controlling the hands. So that's important, but it doesn't mean that, you know, they're obviously, in terms of numbers, a very small proportion, and it sort of behoves us to learn more about the rest.
That is so interesting, because we also use the corticospinal tract as, when we look at tract diameters from the fusion MRI, corticospinal tract is always a reference to see, oh yeah, the largest axons in the cortex tract. So for me, it was interesting to see that distribution where you actually show the majority are small axons.
Yeah,
But
I think it's this is a bit. I mean, this is not new. This has been known for a long time. There's some wonderful studies that were done in the 1930s in the macaque, showing, you know, just how incredibly skewed this distribution is. And initially they thought, oh, well, these fibers must be coming from somewhere else, because they were studying the pyramidal tract. But there's no question that all the fibers that are in the pyramidal tract do come from cortex, and so there's no question that that even the fine ones are also coming from the cortex.
do you have any questions? I see your face, so I want to ask if you have any questions. No. Okay, I might have one last one, if I can keep you one more minute, Roger. Which is, given the coarser, for lack of a better word, representation that we have of the corticospinal tract in tractography, both in terms of definitions and, I guess, cortical terminations and, you know, differentiation within the brainstem, etc. What would be the one thing that you would want to see achieved, let's say, in tractography, light also of its importance within the clinical use of the tract?
Well, I think one, you know, absolutely key area that we need more information about is understanding the difference between the projection that goes to the control brainstem nuclei, including the nucleus ambiguous, and the projection that goes to the spinal cord. This is obviously so important for speech versus hand control. And for instance, you know, in the clinical literature on motor neuron disease, clinicians put a lot of emphasis on whether the disease presents first and foremost as a limb onset problem. So these are people who typically have suddenly realize that their hands are clumsy, or their feet are clumsy, or they can't ski anymore, or something like that. So this is limb onset versus bulbar onset, where they begin to notice that their speech is becoming affected and slurred. Now, those were my own idea is that those involve just two different parts of the projection, one going to the brainstem, one going to the cord. But we don't really know exactly how those fibers travel together, at least as far as the pyramidal tract in the brainstem. So what I'm really saying is we need to understand more about the fine structure of the pyramidal tract itself, how much of the fibers there actually terminate in the brainstem and are involved in the control of speech, and how many carry on and go into the spinal cord. I think that's something that could be done with tractography.
Okay, well, we'll try.
Okay, great. All right. Well, thank you for the invitation.
Thank you very much. This was I've learned a lot. It was wonderful.
Thank you very much.
Okay.
Have a great day.
Thank you, everybody. Thank you.
Everybody for joining. Bye bye. Thank you.
I guess you can stop the recording, and when it's processed, it's gonna get put, on the Zoom account, and I'm gonna tell Chamberlain to cut maybe